focused on a different topic related to antibiotic resistance via enzymes or other key mechanisms involving penicillinase-resistant penicillins, cephalosporins, sulfonamides, fluoroquinolones. Question 1. Enzymatic inactivation of penicillinase-sensitive versus resistant penicillins. A 30-year-old man with cellulitis is treated with nafcillin. This drug is chosen over penicillin G because the organism produces beta-lactamase. What structural modification allows nafcillin to remain effective in this case? A. Methylation of ribosomal binding sites. B. Beta-lactamase inhibitor co-administration. C. Bulky R group that blocks beta-lactamase access. D. Resistance to bacterial acetyltransferase. E. Improved outer membrane penetration. Correct answer. C. Bulky R group that blocks beta-lactamase access. Explanation. Penicillinase-resistant penicillins, e.g. nafcillin, oxacillin, are resistant to beta-lactamase due to a bulky side chain, R group, that prevents enzymatic access to the beta-lactam ring. Question 2. Cephalosporin resistance. A hospitalized patient with a urinary tract infection caused by Klebsiella pneumoniae is started on ceftriaxone. 2 days later, cultures grow ESBL-producing Klebsiella. The resistance is most likely due to A. Mutations in topoisomerase 4, B. Beta-lactamase that hydrolyzes the drug, C. Increased drug uptake, D. Mutation in porin proteins, E. Ribosomal RNA methylation. Correct answer. B. Beta-lactamase that hydrolyzes the drug. Explanation. Extended-spectrum beta-lactamases, ESBLs, can hydrolyze third-generation cephalosporins like ceftriaxone, leading to resistance. This is an enzymatic mechanism of resistance. Question 3. Sulfonamide resistance. A patient with traveler's diarrhea is treated with sulfamethoxazole trimethoprim. Resistance in E. coli is most likely due to which of the following? A. Beta-lactamase production, B. Altered DNA gyrase, C. Mutated dihydropteroate synthase, D. 23S rRNA methylation, E. Ribosomal protection proteins. Correct answer C. Mutated dihydropteroate synthase. Explanation. Sulfonamide resistance is most commonly caused by mutations in dihydropteroate synthase, reducing drug binding and inhibiting folate synthesis. Question 4. Fluoroquinolone resistance via enzymatic target alteration. A 55-year-old man with prostatitis is prescribed ciprofloxacin. The infection persists, and a resistant strain of Pseudomonas aeruginosa is isolated. Which of the following best explains this resistance? A. Beta-lactamase production, B. Methylation of rRNA, C. Mutation in DNA gyrase, D. Inhibition of peptidoglycan cross-linking, E. Inactivation by aminoglycoside modifying enzymes, C. Mutation in DNA gyrase. Explanation. Fluoroquinolone resistance often results from mutations in DNA gyrase, gyr-A gene, or topoisomerase Y4, which reduce drug binding to its enzymatic target. Would you like a visual summary chart of these